We will be presenting a case study about a suspected foodborne outbreak that occurred during a school food fair at SMK Jalan Mulur, Puchong, Selangor. During the event, students bought different foods and drinks from several stalls. Soon after, a number of students visited the nearby clinics with gastrointestinal symptoms such as nausea, vomiting, abdominal cramps, and mild diarrhea. At first, these symptoms could have just been mistaken for overeating or drinking too many sugary drinks, but several students became sick after attending the same event, which raised concern about a possible foodborne outbreak. In this presentation, we will analyze the students' symptoms, food history, possible causes, laboratory investigations, treatment, and preventive measures.
This slide summarizes the profile of the 5 affected students. Patient A, B, D, and E developed gastrointestinal symptoms, including vomiting, nausea, abdominal cramps, and mild diarrhea, while patient C experienced only mild abdominal discomfort and remained clinically stable. The onset of the symptoms ranged from 3 to 12 hours after eating and food exposure. Patients A and E develop symptoms the earliest, within 3 to 5 hours after consuming the food. Fried rice was consumed by 3 of the 5 patients, making it the most likely common food exposure among the affected patients. This slide shows the key clinical findings from the affected students. The most common symptom was vomiting, followed by nausea and abdominal cramps. Some patients also had mild diarrhea. Most patients didn't have fever, so the symptoms mainly involved the digestive system. The onset was quite rapid, appearing within 3 to 12 hours after eating the food and food exposure. Patient E had the earliest onset, while patient D had the latest one. Overall, the main pattern is that student A, B, and E all consumed the food and had more severe symptoms, while patient C didn't and only had abdominal discomfort.
Now, I will explain the differential diagnosis in this case. From the previous slide, we know that vomiting and abdominal cramps are predominant, so we can say it's a foodborne illness, more commonly known as food poisoning. Now, food poisoning can be toxin mediated or infection mediated, but infection mediated ones are more commonly associated with the longer incubation period and fever. Since in this case there was no fever and the incubation period was short, we can say that it was a toxin mediated food poisoning. So, our first differential diagnosis was the Bacillus cereus food poisoning, particularly the emetic type. This organism was considered because several affected students had consumed fried rice, which is a well known vehicle for Bacillus cereus food poisoning. In addition, the onset of symptoms occurred within a few hours after food consumption, and vomiting was predominant symptom. The second possible cause was Staphylococcus aureus food poisoning. Similar to Bacillus cereus, it produces a performed interoxin that can cause rapid onset nausea and vomiting within a few hours of ingestion. The absence of fever and predominance of vomiting also support this possibility. Finally, our third differential diagnosis was Clostridium perfringens food poisoning, as it is commonly associated with improperly stored cooked foods. However, this pathogen typically presents with abdominal cramps and diarrhea rather than severe vomiting. Now, for the most likely diagnosis, let's see the flowcharts. We first identified this as a foodborne outbreak since multiple students developed gastrointestinal symptoms after attending the same food fair. The next key clue was the rapid onset of symptoms, with most patients becoming ill within 3 to 8 hours after eating. This suggested a toxin mediated foodborne illness rather than an infection mediated illness, which typically has a longer incubation period. We then examined the clinical presentation. Vomiting was predominant while diarrhea was mild or absent, and most importantly, none of the patients developed a fever. These findings further supported a toxin mediated illness. Another significant epidemiological clue was that several affected students had consumed fried rice, including patient E, who reported eating only fried rice before developing a severe vomiting within 3 to 4 hours. Since fried rice is a well recognized vehicle for emetic Bacillus cereus food poisoning, we suspected that was the cause. We also considered other possible pathogens like Clostridium perfringens, but that was less likely because it typically presents with diarrhea, and diarrhea was absent in this case. For Staphylococcus aureus, it was a reasonable differential diagnosis due to the similar rapid onset and toxin-mediated presentation. However, the strong epidemiological association with fried rice made Bacillus cereus the better fit overall. Therefore, based on the available evidence, Bacillus cereus food poisoning emetic type is most likely the diagnosis. Nevertheless, laboratory confirmation through food and clinical specimen testing would still be required to establish diagnosis definitively.
The food at this food stall might have been contaminated with Bacillus cereus, which when introduced to the food grows and multiplies at room temperature and produces cellulite, a heat-stable enterotoxin. Patients A, B, and E generally complained of abdominal cramps, nausea, and vomiting. When the food is consumed, in this case fried rice, the cellulite is ingested by the person. The cellulite is able to survive the acidity and digestive enzymes, the gastric barriers, where it is absorbed into the blood through intestinal capillaries. The cellulite is then distributed throughout the body and binds to 5-hydroxytryptamine 3 receptors in the stomach and intestine to cause mitochondrial dysfunction. This leads to the activation of vagus afferent nerves, which in turn activate the vomiting center in the brain, leading to nausea and vomiting. Mitochondrial dysfunction also impairs the intestinal barrier and the epithelial mucosa, causing inflammation, which then disrupts the normal fluid balance of the intestine, leading to diarrhea. There were also complaints of abdominal cramps, which is because cellulite interacts with the neural pathways of the gastrointestinal system. As for patients C and D, they did not consume fried rice, but rather consumed fried noodles and sandwiches. While their symptoms were not as severe, they also reported some symptoms. The variation in these symptoms has to do with the fact that cellulite causes dose-dependent effects, meaning if a lower concentration of the toxin was ingested, the symptoms would be fewer and less severe. This can also be observed in patients A, B, and E, because some patients reported the nausea and vomiting to be more severe or repeated.
To investigate the foodborne outbreak, clinical samples such as stool and vomit are collected from symptomatic students. Leftover food, including fried rice, and environmental swabs from food preparation areas are also collected to identify the source of contamination. The samples are transported under refrigeration, stored at 4 degrees Celsius, and prepared for testing by homogenization and serial dilution. They are then cultured on selective media to detect the suspected bacteria. Once bacterial colonies grow, confirmatory biochemical tests are performed to verify their identity. Finally, toxin detection is carried out because food poisoning symptoms are caused by bacterial toxins rather than the bacteria alone. Detecting toxins such as cellulite from Bacillus cereus, enterotoxins from Staphylococcus aureus, and Clostridium perfringens enterotoxin helps confirm the causative pathogen and supports the outbreak investigation.
Moving on to our diagnostic results shown. First, looking at the HbA plate, our culture produced large, dull, gray-white opaque colonies featuring rough wavy edges and a broad zone of beta hemolysis. When we subjected these colonies to a gram stain, they tested positive, appearing as distinct purple-blue cells. We can observe them arranged as streptobacilli, and we may also notice the presence of ovoendospores. For biochemical phase, the VP test was positive, yielding a characteristic pinkish to red deep color change. Finally, the ELISA test returned a negative result compared to other toxins, indicated by a lack of yellow or blue color change in the well. Based on our lab investigations, the results point directly to Bacillus cereus. For the treatment plan, primary management focuses strictly on supportive care, specifically oral rehydration therapy to counteract severe fluid loss. We also recommend dietary modifications. This involves resting the stomach for a few hours after vomiting stops, followed by a gradual introduction of bland foods using the standard brat diet. This includes bananas, rice, applesauce, and toast. Looking at our medication strategy, antibiotics are actually not recommended. This is because the illness is driven by preformed heat-stable toxins rather than an active invasive infection. Antibiotics won't alter the course of the toxin's effect. Antiemetics can be considered if the vomiting is severe enough to prevent oral rehydration therapy, though they are usually unnecessary as vomiting typically subsides quickly.
To control the outbreak, first to stop serving the suspected food, especially fried rice, to prevent further exposure. Leftover food should be kept for lab testing to confirm the contamination source. Food history, symptoms, and on-site time should also be verified across all affected students to identify the common exposure patterns. Symptomatic students should be isolated and referred for medical care to manage dehydration caused by repeated vomiting. The outbreak must also be reported to health authorities so proper investigation and control measures can be carried out. For prevention, strict food hygiene practices are essential. Proper hand washing and clean surfaces help reduce contamination from organisms such as Staphylococcus aureus. Raw and cooked food should also be separated to prevent cross-contaminations during preparation. Food must also be cooked thoroughly, and most importantly, cooked rice should be stored properly and not left at room temperature for more than 2 hours, as this allows Bacillus cereus to produce toxins that lead to rapid vomiting.
In conclusion, this case is most likely a foodborne outbreak caused by Bacillus cereus emetic type food poisoning, with fried rice as a suspected source. This is supported by the rapid onset of symptoms, vomiting as the main complaint, absence of fever, and the fact that several affected students consumed fried rice. Patient E is especially important because they ate only fried rice and developed severe vomiting within just a few hours. Although other cases such as Staphylococcus aureus and Clostridium perfringens were considered, they fit the case less strongly. Overall, this outbreak highlights the importance of early recognition, reporting, proper food storage, hygiene, and temperature control to prevent further illnesses and protect student health during food events.
Throughout this case study, we learned that completing a microbiology presentation is not just about understanding the science, but also about working together as a team. Each of us were responsible for different sections, and although everyone focused on their own part, we had to make sure the presentation flowed logically from one slide to the next and was cohesive. This meant communicating regularly, discussing our findings, and making sure our explanations supported one another. One of our biggest strengths was our ability to research information from reliable scientific journals and compare different sources before deciding what to include. Rather than copying everything we found, we worked together to identify the most relevant points and present them in a way that was accurate, concise, and easy to understand. We also became better at summarizing complex microbiology concepts into presentation ready content without overwhelming the audience. Throughout the process, we supported each other by giving feedback on the slides, on the recordings, checking the references, and making sure everything was visually organized and consistent. At the same time, we faced several challenges. Since many of our assignments had similar deadlines, managing our time effectively was one of the biggest difficulties. We also had to ensure that everyone had completed their sections on time so the presentation could be reviewed as a whole. Another challenge was deciding how much information to include. There were many research articles available, but not everything was relevant to the case. We had to carefully balance scientific information with the presentation time limit while making sure our explanations remained accurate and detailed. This case study itself also challenged our thinking. We had to compare different foodborne pathogens, interpret the patient's symptoms and incubation periods, and justify why Bacillus cereus was the most likely causative organism instead of other possible pathogens. This taught us to evaluate evidence critically rather than making assumptions based on a single finding. It also showed us the importance of considering both the clinical presentation and the epidemiological evidence before reaching a diagnosis. Overall, this project strengthened our research, communication, teamwork skills while improving our confidence in presenting scientific information. More importantly, it helped us understand how microbiology knowledge is applied in real outbreak investigations through systematic analysis and evidence based reasoning. In future projects, we hope to manage our time more effectively by starting earlier, allowing more time for discussion and practice before recording. We also want to continue improving our ability to communicate scientific information clearly and work collaboratively, as these are essential skills for future laboratory work, research, and clinical microbiology practice.